Hello, this is Dr. Lauren Launen, and this is a lecture about translation. The purposes of this lecture are to discuss the structure of a ribosome with attention to the presence and role of ribosomal RNA, and the differences and similarities between prokaryotic and eukaryotic cells. We'll talk briefly about the 3 stages of translation. We'll also learn one practical in the context of medical application that takes advantage of the difference between prokaryotic and eukaryotic ribosomes, and we'll learn 2 factoids of scientific information that pertain to ribosome structure and function. So what do ribosomes do? Ribosomes are responsible for the catalysis of the peptide bond formation, and what that means is they take 2 amino acids, so here we've got an amino acid here, an amino acid here shown in blue and red, and they form a peptide bond. This is the peptide bond here. So the peptide bond is connecting the carbon within the carboxyl functional group of this amino acid with the nitrogen that was in the amino group of the neighboring amino acid. And this structure is referred to as a dipeptide. Once you have many amino acids, you use the term polypeptide. This in blue is considered one amino acid residue, and here in red is another amino acid residue. That's the term that we use here. Within a peptide, there's an orientation or an endedness. There is an N terminus, which right here are amino terminus, and there's a C terminus, which is shown right here, also called the carboxy terminus. So the formation of this peptide bond is accomplished by the ribosome, and the ribosome also facilitates the work of transfer RNA, which helps to carry these amino acids into the ribosome, and the transfer RNA will base pair through the anticodon region with the messenger RNA, which will also be entrapped in the ribosome, and together that will accomplish the correct selection of the appropriate amino acid. Translation is one stage in gene expression. We have already discussed in class the structure of a gene, and we have talked about how transcription is the process of using the template strand of a gene in order to make corresponding messenger RNA. Now we're talking about what happens to that messenger RNA as it is utilized to make a polypeptide, shown here, this step here. And this happens in the ribosome. It's done by the ribosome. So here's a photo of a ribosome, or a cartoon more accurately, of a ribosome, showing in blue the large subunit and in purple the small subunit. Feeding through the ribosome in this bright green color is the messenger RNA. And inside the ribosome, when it is assembled on the messenger RNA strand as shown here, there are 3 pockets that are connected by channels. These pockets are referred to as the A, the P, and the E site. And the ribosome in this cartoon is traveling in this direction, okay, towards the right, and down, following the messenger RNA. These little boomerang-shaped molecules are the transfer RNA molecules, which we have discussed earlier in class. Each transfer RNA molecule is a shape that is such that it allows it to fit within the pocket or pockets of the ribosome. So this little transfer RNA molecule is going to be able to come into the A site, and it will base pair between the anticodon region here and the codon region on the messenger RNA. So here, if you look in carefully, you'll see anticodon, codon, base pairing. That's how this amino acid gets into the ribosome in the first place. Here, coming out of the large subunit, is the polypeptide. So each of those little 3-letter names or nicknames are the shorthand for amino acids. So PRO is proline, ARG is arginine, and so on. Here's the N terminus. That's the part that comes out of the ribosome first, so sort of hidden in here. At this end is the C terminus. Okay? So this ribosome is actively translating. It's doing this step that we refer to as extension, meaning it's extending the polypeptide. And so what will happen is the ribosome will move one codon of messenger RNA at a time, and as that happens, whatever tRNA molecule is at the A site will be bumped to the P site, whatever was at the P site will be bumped to the E site, whatever was at the E site will be bumped out. And notice that it is a discharged non-amino acid carrying tRNA, it will go off and encounter the enzyme aminoacyl tRNA synthetase or synthase, which will replace this spot here, recharging it with another amino acid. So the two major things that the ribosome is accomplishing is the formation of peptide bonds, which is a catalytic activity, and ensuring the correct choice of amino acid, and that choice is mediated by tRNA, mRNA, anticodon-codon base pairing. All right, so a little bit about ribosomes. The ribosomes are found in all cellular life forms, and they have a very similar structure in all cellular life forms. However, there are some differences between eukaryotic ribosomes, such as what is found in our own cells, and those found in prokaryotic cells, the cells that belong to the domains archaea and bacteria. So shown here is an intact ribosome from a eukaryote, for example, a mammalian ribosome, and it is 80 S in size. The S is a unit that is the Svedberg coefficient, or Svedberg unit, and it is determined by allowing these ribosomes, or any material at all actually, settle in a thick solution, a thick sugary solution, and then you measure how long it takes for them to settle, and you use an equation and figure out the mass or the size, and it's a relationship of mass and also surface area. The large subunit of a eukaryotic ribosome is 60 S in size. The small subunit is 40 S. You'll note that 40 plus 60 do not equal 80, but when you put these two things together, the surface area is, of course, not additive, and so that affects or changes the Svedberg coefficient. Each ribosomal subunit is composed of a mixture of ribosomal RNA, or rRNA, and also ribosomal proteins. So the 60S subunit has 49 ribosomal proteins and 3 molecules of ribosomal RNA. The small subunit has 33 ribosomal proteins and 1 molecule of ribosomal RNA. So I'm going to point out a couple of important features here. This molecule, the 18S rRNA found in the small subunit, is responsible for the initiation of translation. It plays a key role that I'll talk about in a moment. This molecule, the 28S rRNA molecule, is what is largely responsible for the peptide bond formation, the catalytic activity of a ribosome. Now here's a prokaryotic ribosome, and it's oriented differently, so it's sort of tilted on its side, but the principles still apply. Prokaryotic ribosomes are smaller, so this is the 70S ribosome. The large subunit is 50S, and the small subunit is 30S. In the large subunit of a prokaryotic ribosome, there are 2 molecules of ribosomal RNA and 31 ribosomal proteins. In the small subunit, there are 21 ribosomal proteins and 1 molecule of ribosomal RNA called the 16S. The 16S rRNA molecule, like the 18S rRNA molecule, is responsible for helping out during the initiation of translation. And the 23S, like the 28S in the eukaryotic ribosome, is responsible for catalyzing the formation of peptide bonds. So we call that catalytic activity peptidyl transferase activity, or peptide bond forming activity. And because this is an activity that is mediated not by a protein enzyme, but instead by an RNA molecule, we call it not an enzyme, but a ribozyme. So an interesting factoid is the fact that there are some really practical applications to really understanding ribosomal structure. So one of the things that we search for when we are trying to develop new antibiotics that target a bacterial pathogen that is infecting a human eukaryotic body, we look for differences between the cellular structure and function between the pathogen and the eukaryotic host. And the reason we look for differences is it is our goal in drug design when we're making antibiotics to find a drug that will target and damage the pathogen but not harm the host. This is something called selective toxicity. And so it turns out that although prokaryotic and eukaryotic ribosomes are incredibly similar, the subtle differences between the two, and I just showed you images showing that difference in part, based on size at least, those subtle differences can be capitalized on, and in fact about half of the antibiotic drugs that we use to treat bacterial infections target bacterial ribosomes, and specifically they target the peptidyltransferase activity center, which is deep here within the large subunit of a prokaryotic ribosome. This is a prokaryotic ribosome, okay? And so these are antibiotic drugs, so that's virginomycin, carbamazine, etc. A little bit hard to see them because the font is quite small, but these are antibiotic drugs, different ones that you might take to treat an infection. And what they will do is they will bind with the ribosomes of bacterial pathogens, and they will stop them from functioning. Those bacterial pathogens, when they can't make proteins anymore, they will die. And so when they die, you've effectively treated the infection. So I'm going to stop this first half of the lecture on translation, and you will then go to the next lecture to complete this lecture series.